Dear student, I am Dr. Prakash Mungli, a professor of biochemistry. In this video, I will be explaining you about bioenergetics. Let's get into that. Bioenergetics, it is energy changes in the chemical reaction. So, our biological system, it is isothermic. We are isothermic animals. That means we maintain a constant temperature in our body. That means we maintain a temperature about 37 degrees centigrade. Why this 37 degrees centigrade is important for us? Because at 37 degrees centigrade, all our enzymes will reach optimum activity there, and the optimum activity of an enzyme will make sure our biological reactions runs in an optimum direction so that metabolic pathways will go on properly and cellular function will be going on in a proper way. So, the chemical energy that powers biological process has to come from the food that we consume. Because when we consume food, so when we digest the carbohydrates, proteins, lipids, these are the macromolecules that are present in our food. So they will undergo catabolism, and when a food molecules like carbohydrates, proteins, lipids, when they undergo catabolism, there will be release of energy, and that chemical energy, it will power biological reactions that will be going on in our body for cellular metabolism. So, if there is any energy imbalance in our biological system, it will lead to metabolic abnormalities. So, what are the hormones that will maintain our energy metabolism, or what are the hormones that controls energy metabolism? So, the important hormones that will control energy metabolism is they are insulin, glucagon, epinephrine and thyroid hormones. These are the hormones that will control our metabolic pathways, and thereby they control energy metabolism in our system. If there is any lack of chemical energy, like what we see during starvation, if there is a starvation, that means when we don't consume, when the person do not consume food, that means the food which has to bring that chemical energy into the system, that will be decreased, and that creates an imbalance, means lack of energy, that will decrease the biological processes, and that might result in death. And the other way around, if there is any excess energy, so the excess energy may also result in metabolic abnormalities, and it will give rise to overweight and obesity. So, on either direction, whether it is a lack of energy or the excess energy, both of them may lead to metabolic abnormalities. That is why it is important for us to maintain energy balance in the system. Now, let's get into a concept called delta G. Delta G, it is the energy that is available to do work. So, it means it is the energy difference between the substrate and the product. Whenever a substrate is converted to product, either energy is getting into the reaction or energy is coming out of the reaction. So, the difference in the energy between the substrate and product, we refer that as the free energy of the reaction, and this is the energy that is available to do work. So, whenever and it is difference in the energy between a substrate and product, we refer it as a delta G there, and delta G, as it approaches, when the value of delta G approaches to 0, it will lead a reaction into equilibrium. And also, value of delta G, it will predict a reaction, the direction of the reaction, it will be predicted if we know the value of delta G. So, it will predict whether the reaction is favorable or the reaction is not favorable. Okay, while calculating delta G, so delta G, the formula for calculating delta G is delta H minus T delta S. So, delta H here, it is enthalpy, and that is heat that is released or absorbed during the reaction. The difference in that heat, we measure it as delta G, and delta G do not predict whether reaction is favorable or not. Then we have something called as delta S. Delta S is the change in the entropy. It is the measure of randomness of the reaction. Higher the randomness in the substrate, so it is converted into product. Lesser the randomness, So the conversion of substrate into product will be decreased. So again, just delta G do not predict whether reaction is favorable or not. But when you combine these parameters together, where delta G equals to enthalpy minus temperature and entropy. So together when we get value, if the delta G value, if it is 0, that is an equilibrium reaction, and based on the sign, whether positive or a negative sign for delta G we get, so that we can predict a reaction as we depending on the delta G value there. Now, the free energy, it is the energy that is available to do work. So we can measure that free energy with the delta G, which is referred as Gibbs free energy. Gibbs change in free energy is the useful energy in the system to do work, and as we have seen in the previous slide, we can measure free energy as delta G equals to delta H minus T delta S, and this delta G also provides a measure of energetic feasibility of a chemical reaction. Now let's look into the details how a substrate is converted into a product. Now, as you see here in this particular figure, a substrate A is converted into a product B. Now look at the energy level of free energy level of substrate, which is at this point here, and look at the free energy level of the product, which is below here. So that means a substrate which is at higher energy level is forming a product which is at lower energy level. So basically, substrate which is at higher energy level, it is forming a product at the lower energy level. That means there will be release of an additional energy, because substrate is coming from higher to lower, right? That means any molecule that has higher energy, it is a high unstable molecule. So unstable substrate here, that is A, means reactant A is going to become product B, which is relatively more stable because it is at lower energy level. Okay, so in order, before it goes to the lower energy level final product, it has to reach a highest energy level. That's what is shown here, and that highest energy level of the reactant is called as a transition state of the reaction. So when it reaches highest energy level, from there it falls down into a product which is at lower energy level. Now let's look at the energy difference between the reactant and the product. Reactant A and the product B, and the energy difference there. See, here it is. It is initial state and the final state. So the difference in the energy between initial state and the final state is basically referred as delta G. That is, it is the energy difference between the reactant and the product. Now, because the reactant, which is A, which is at higher energy level, and product is B, lower energy level. So when A is coming to B, so the energy that is there in A, which is at higher energy level, as it is falling down to the lower energy level, so the additional energy that was there, it is released into the system, and that makes delta G negative. And whenever delta G is negative, it indicates this type of reactions are spontaneous reaction, because there is sufficient energy in the reactant to make a product which is at lower energy level. So that is when we see delta G negative. So whenever you see delta G value in negative numbers, it is a spontaneous reaction, and if the delta G negative value is at higher number, so that basically it is an irreversible kind of reaction. Now let's look at number B here, figure number B. So where the if you look at the initial state of the reactant, that is B, which is at lower energy level, and reactant A, which is reaching at higher energy level. Basically, lower energy substrate is reaching at higher energy product. That means energy has to be given to the system, because delta G becomes positive here, because lower energy substrate going into higher energy product. So in order to take it there beyond the energy that was there in the substrate to highest energy, and then into a certain state where still it is much higher than the initial state of a molecule. So that particular type of reactions will show delta G positive. So this type of reactions are endergonic reactions. That means delta Z value will be positive. Higher the positive delta Z value, so higher the reaction becomes, more endergonic, and feasibility of the reaction becomes difficult unless we provide external energy into the system. Now let's look at the sign of delta Z, which will predict direction of the reaction. If the delta Z is in negative number, as I told you before, delta Z is negative, that is less than 0, delta Z is less than 0, that is in negative number, the reactions are spontaneous and these are exergonic reaction and the additional energy is released into the system. If the delta Z is in positive values, delta Z is more than 0, that is positive number, so this type of reactions proceeds only if additional energy is added into the system, and these are endergonic reaction. And if the delta Z is equals to 0, and that type of reactions are equilibrium reaction. Remember this. So delta Z negative means spontaneous, exergonic. Delta G positive means these are non-spontaneous reaction, endergonic reaction. Delta G 0 means it is an equilibrium reaction. Now here in this table, we are comparing delta G here and the rate of reaction. So delta G is of an enzyme is not delta G is not affected by enzymes because enzymes do not change delta G. We are going to see that in enzyme lecture. So if delta G is less than 0, thermodynamically these are spontaneous reactions and energy is released into the system, and usually this type of reactions are irreversible. Delta G positive means thermodynamically these are non-spontaneous reactions, these are endergonic reactions, energy is required to make the reaction feasible. If delta Z is equals to 0, reaction is at equilibrium, that means these are freely reversible reactions. If delta Z dot here is the standard free energy change at a specific concentration. Now, enzymes, what they will do? Enzymes, they will increase the rate of reaction by simply decreasing the free energy of activation, which is referred as delta G++. We are going to see how exactly this is done by the enzymes in enzymes lecture. Alright, in a biological reaction, when a reaction where the substrate is converted into product, so there is a change in energy between substrate and product and we measure it as delta G. Let's take an example of conversion of glucose into fructose 6-phosphate. Okay, so in that very first reaction here, number 1, glucose conversion into glucose 6-phosphate. So basically the phosphate is added at the 6th carbon of glucose here, and the delta Z here for conversion of glucose to glucose 6-phosphate is plus 3.3 kilocals per mole. You need to take a look at that plus number, positive number. It's an endergonic reaction because delta G is positive. That means energy has to be given into the system to make this reaction possible. Otherwise, conversion of glucose to glucose 6-phosphate, it's thermodynamically a non-favorable reaction. It will take time. So, in order to enhance the rate of reaction here, energy has to be supplied into the system because we need conduction of this reaction needs plus 3.3 kilocals of energy. How that is made possible? So, whenever a reaction is endergonic, always remember in a biological reactions, whenever a reaction is endergonic, there comes ATP. ATP is the energy currency of our body, of our cells. Whenever there is a need of energy, ATP breaks into ADP plus inorganic phosphate, or ATP simply breaks into ADP and inorganic phosphate can be used for a reaction. Like here, glucose is converted into glucose 6-phosphate by breaking ATP into ADP and phosphate is added to 6th carbon of glucose. And when ATP breaks into ADP, amount of energy that is released at that time is minus 7.3 kilocals of energy. That means breakdown of ATP into ADP, it is a thermodynamically spontaneous reaction. It's an exergonic reaction. Minus 7.3 kilocals of energy is released into the system, and that energy can be used to conduct endergonic reactions like conversion of glucose into glucose 6-phosphate. So that means what happened here, we needed 3.3 kilocals of energy, and we got minus 7.3 kilocals of energy by breaking ATP into ADP. So minus 7.3 kilocals there, and take plus 3.3 from that minus 7.3. Basically you subtract 3.3 from that minus 7.3 kilocals. You don't worry about minus and plus here, it is just indicating exergonic and endergonic kind of reaction. So we have 7.3 kilocals. Imagine that you have $7.3, $7.30. So out of that you need to spend $3.30, right? So that means if I take $3.30 from $7.30, that means I am going to get around $4 left with me. Still it is in minus sign. So minus 4 kilocals of energy still there in the system, and this minus 4 kilocals of energy can be used to conduct further reaction, as long as the energy required for conducting next reactions are not more than minus 4.0 kilocals. So let's see what is the next reaction. Next reaction is conversion of glucose 6-phosphate into fructose 6-phosphate. For this reaction, there is a need of plus 0.4 kilocals. Only 0.4 kilocals are needed, but how much energy that is left in the system here? It is minus 4.0 kilocals of energy is still there in the system. That is $4 is there. All you need is only 40 cents. So you can take that 40 cents from the $4. So what is remaining now is, if you take out 30 cents there from $4, what is remaining is $3.60, and that is minus $3, minus 3.6 kilocals. Dollar I am just saying just to correlate the number, just to make it easy. Otherwise, my point here is, as long as that you have that energy in the system, and as long as the endergonic reactions needs energy less than what is available in the system, these reactions can go on by using the available energy. So although these are endergonic reactions, but these reactions are made possible by breaking ATP into ADP and releasing the energy into the system, and that energy can be used by any other reactions which are endergonic. It's basically what we are doing here. We are conducting, the cell is conducting an exergonic reaction, release energy into the system, and make sure this particular energy is used by some of the reactions that needs energy, which are endergonic, which needs energy, and that energy used by the exergonic reaction. It's a kind of coupling reaction. So like this, conversion of glucose into fructose 6-phosphate is done by breaking ATP into ADP, making the system exergonic, and using making possible some of the endergonic reactions are still conducted. At the end of the conversion of glucose into fructose 6-phosphate, after using one ATP, still we system has minus 3.6 kilocals of energy. From now the next reaction, that is if the fructose 6-phosphate goes into another reaction like conversion of fructose 6-phosphate into say fructose 1,6-biphosphate or any other reaction, as long as the energy required for that reaction is less than 3.6 kilocals, reaction is made possible. If the energy required for the next reaction is more than 3.6 kilocals, like if it is plus 5.0 kilocals, that reaction won't be possible. Why? Because we have only minus 3.6 kilocals, and energy required for the next reaction it should be either 3.6 or less than that. If it is more than this, reaction won't be possible. That means we need to break one more ATP, and that's how the reactions will be made possible in biological reaction. So now let me sum it together. It is metabolic pathways that run in forward direction when overall sum of delta G is negative, even if some of the reactions in the pathway is positive. That means, as you see here, some of the reaction in these pathways, in the conversion of glucose into fructose 6-phosphate, like first reaction and the third reaction, they were positive, means endergonic. But since the conversion of ATP into ADP was exergonic, minus 7.3 kilocals, so these reactions are made possible. Still we have minus 3.6 kilocals of energy left in the system, and that's how the metabolic pathways run. So in a metabolic pathway, whenever there is an endergonic reaction, cell is going to break ATP and make sure the reactions will go on in a forward direction. That is an important point that you should remember to understand metabolic pathways. Alright, now when endergonic reaction is there and when there is a need for energy, so the endergonic reactions can be simply coupled with the exergonic reaction. Any reaction that releases energy, that energy can be coupled with the endergonic reaction that needs energy. This particular simple diagram here, it is going to explain you how in a metabolic pathways these coupling reactions can go on. As you see here, conversion of a reactant C into product D, it's an endergonic reaction here. Okay, and that means there is a need for energy here. Delta Z is positive. How to make this reaction possible? So we need to couple a reaction which is exergonic. In the figure, conversion of A to B, where A is at higher energy level and B is at lower energy level. So the energy is released into the system, and this energy is coupled with endergonic reaction, which is reactant C going from lower energy level to a product D going at higher energy level. So basically coupling is done here. So A to B reaction is coupled with C to D reaction. So with that coupling, reaction is made possible. I am going to rewrite in a different way here. Consider a conversion of A to B is endergonic, that is delta Z equals to plus 4.0 kilocals. Okay. Now it is an endergonic reaction. So how to make this reaction possible? So I am going to break ATP. I am going to couple a breakdown of ATP with this reaction, ATP into ADP plus PI. So that particular reaction gives delta Z minus 7.3 kilocals. So minus 7.3 kilocals of energy is coupled here with a reaction that needs plus 4.0 kilocals. So this reaction is made possible because we have higher energy that is coming from exergonic reaction, that is breakdown of ATP into ADP, and I am going to means that particular reaction is coupled with the conversion of A to B. So endergonic reaction is coupled with exergonic reaction. So that type of coupling of reactions is made possible in a biological system to complete the process. So overall, in biological system, oxidative pathways, which are catabolic pathways, referred as catabolic pathways, are always coupled with reductive biosynthesis pathways, referred as anabolic pathways. So oxidation coupled with reduction. So whenever there is oxidation going on, oxidation pathways will release energy. So these are catabolic pathways, and these catabolic pathways, the energy coming out of the catabolic pathways are used to connect that with the anabolic pathways, which are reductive biosynthesis pathways, are usually endergonic pathways. So exergonic catabolic pathways, endergonic anabolic pathways, both are coupled here. Let me write it down. So catabolic pathways or exergonic. These are exergonic pathways, and anabolic pathway is a endergonic pathway. So basically exergonic is coupled with endergonic. So the energy coming out of exergonic pathway will be coupled with endergonic pathway, that is anabolic pathway. That is how our biological system will run. So that is what is shown here in this particular figure. As you see, conversion of C into D is an endergonic reaction. Conversion of B into A is an exergonic reaction. So energy that is released in the exergonic reaction is coupled with the endergonic reaction and made the reaction possible. So transfer of free energy from exergonic reaction to endergonic reaction via high energy intermediate compound is shown here. Now, one of the things that you need to remember in the figure here is wherever we mention high energy bond, and that particular bond is mentioned with the symbol like this. This is the high energy bond symbol. Okay, so whenever you see that particular symbol, So remember, it simply indicates a high energy bond there. Now, let's look at high energy intermediates in a biological system. So, in a biological system, endergonic reactions are not directly coupled to exergonic reactions, but via molecule with a high energy potential. And one of the important molecule, the principal high energy intermediate in our cell, which is referred as energy currency of our cell, it is called as adenosine triphosphate, simply written as ATP. ATP is the energy currency of our cellular metabolism. Okay, now let's look at some of the energy molecules in this particular figure. So we have phosphoenol pyruvate. Look at the amount of energy that is present in phosphoenol pyruvate. It is minus 14.8 kilocalories per molecule. And compare that with the ATP, breakdown of ATP into ADP. Okay, ATP into ADP, there will be release of minus 7.3 kilocalories of energy. Now, compare this minus 7.3 kilocalories of energy with the phosphoenol pyruvate. The amount of energy present in the phosphoenol pyruvate, it is minus 14.8. That means almost double amount of energy present in phosphoenol pyruvate. Like that. So, any molecule that is giving energy more than that of ATP, including ATP, any molecule that gives energy more than that of ATP is referred as high energy molecule. Okay, so that means we have phosphoenol pyruvate, carbamyl phosphate, 1,3-bisphosphoglycerate, creatine phosphate, breakdown of ATP into AMP. All of them, they show higher energy than that of ATP. These are high energy molecules. Some of the molecules which will give energy lower than that of ATP, it's like glucose 1-phosphate, breakdown of inorganic phosphate, fructose 6-phosphate, glucose 6-phosphate and glycerol 3-phosphate. They are all low energy molecules. Alright, now let's look at the amount of energy that is coming from breakdown of ATP. As I already shown you in the previous slide, it is minus 7.3 kilocalories of energy. So, any molecule that gives energy higher than this number, they are simply called as high energy phosphates or high energy compound. Any molecules that gives energy less than this number, minus 7.3 kilocalories, they are simply referred as low energy phosphates. And this energy, these energy phosphate, high energy phosphates act as energy currency of our cell, and most important energy currency of our cell is ATP, adenosine triphosphate. Okay. Now here in this figure, so one of the high energy phosphate, that is creatine phosphate, it is the reaction of creatine phosphate is shown here. As you can see, there is a high energy phosphate bond that is mentioned with the high energy phosphate symbol there, and the creatine kinase enzyme, it is going to break creatine phosphate into creatine. Okay, so basically ADP in this reaction will be converted into ATP. That means high energy phosphate that is present in creatine phosphate, it will be used to convert ADP into ATP. So there is a formation of ATP here, and this ATP, it has got energy minus 7.3 kilocalories of energy, and also breakdown of creatine phosphate here into creatinine, it is giving minus 12.6 kilocalories of energy per mole. So now there are three major sources for high energy phosphates in our body, and the most important source of high energy phosphate in our cells, which is making ATPs, is the electron transport chain, ETC, which is also called as oxidative phosphorylation. Oxidative phosphorylation is also referred as electron transport chain, and in electron transport chain there is synthesis of ATP, and ATP is a high energy phosphate molecule, and that is one of the major source of high energy phosphate for us in our cell, and that's why ATP is referred as energy currency of our body. Not only oxidative phosphorylation, of course oxidative phosphorylation or electron transport chain is the major source of ATP for us in our cells. Other than that, we have glycolysis, breakdown of glucose into 2 pyruvate molecules, and these 2 pyruvates can further undergo oxidation into acetyl-CoA, acetyl-CoA can undergo further oxidation. During this process, there can be generation of ATPs in the substrate level, in the cytoplasm itself, which is called as substrate level phosphorylation. So, in the glycolysis, substrate level phosphorylation, substrate level phosphorylation in glycolysis will give ATPs in the cytoplasm itself. That is another source of high energy phosphate. And also the citric acid cycle, which is going on in the matrix of mitochondria, which is oxidizing acetyl-CoA into 2 carbon dioxide molecule, will also generate high energy molecule like GTP, guanosine triphosphate. So, that is also a source of high energy molecule there. So, there are 3 major sources for high energy molecules in our cell, and the most important one is oxidative phosphorylation, that is electron transport chain. Second is glycolysis, where substrate level phosphorylation is going on, and the third is citric acid cycle, where a substrate level phosphorylation is going on and generating high energy phosphate there. So, phosphogenes, let's look at briefly about what all these phosphogenes. Phosphogenes, they act as storage forms of high energy phosphates, and it includes creatine phosphate, which occurs in the skeletal muscle, heart, spermatozoa and brain. So, it stores the energy there. So, that is what is shown in the figure. Previously, I have shown you creatine phosphate, which has stored energy here as high energy phosphate that can always be broken down to convert ADP into ATP. That's how a cell can synthesize ATP and use that ATP for whatever the energy needs of cell is there at that time. And creatine phosphate you are going to see in skeletal muscle, heart, spermatozoa and brain. It is the storage form of energy for us. So, phosphogenes, they balance ATP-ADP ratio in cells and in the tissues whenever there is a need for that. So, that's about bioenergetics, and we have seen the different types of reactions and the delta G values, whether and how delta G value can determine whether the reaction is spontaneous, reaction is non-spontaneous, exergonic, endergonics, and then we have seen different types of energy molecules, high energy molecules, low energy molecules, how endergonic reaction is coupled with exergonic reaction, how a catabolic reaction is coupled with anabolic reactions, and that type of energy changes we have seen in this particular lecture. Thank you.